So we're looking at a sarcomere. And let's remind ourselves exactly what we're looking at. So here's a muscle. All right. If we took a cross section of this muscle, we would see many muscle fascicles. If we took a cross section of a muscle fascicle, we would see many muscle fibers or muscle cells. And if we took a cross section and looked even deeper inside a muscle cell, we would see many myofibrils. Here's a myofibril. And if you looked at a myofibril, this is what gives the muscle its striated appearance, remember the myofibril, we would see on this myofibril many repeating units. These are called sarcomeres. Okay. So here's a muscle, let's say a muscle body. Here is a fascicle. Here is a fiber. Remember, fiber is synonymous with a muscle cell. Here is a myofibril. And I apologize if that's super small. And then this is a single sarcomere. So let's look at one sarcomere and look at the structures of these proteins. Let's make that a bit smaller here. All right, so along the center, we've got the thick filaments here. These are joined together by the M line. We'll label these out in a bit. And then we've got the extension of these thick filaments. Let's just make this. All right, and let's put this in a different color. So we've got the myosin filaments. So these are these extensions of myosin, essentially. Okay, now let's draw in actin, or the thin filaments. So these are going to be out to the periphery of the sarcomere. So we've got one, on either side here. Okay. And so basically this is the Z line. So here's a Z line. Here's our other Z line. Okay, remember these are formed by actin. So actin filaments are joined together via the Z line, and one Z line to another Z line is a sarcomere. Let's actually write that out at the bottom here. So a sarcomere is bordered by two Z lines. Okay. Let's actually label a couple things out here. So we've got actin. So here's actin. Okay. Here's myosin. Here we said is the Z line that connects the actin filaments. Here we said is the M line. This is what connects the thick filaments along the center. So here's the M line. Now we can think about the bands or the zones. So the zones or the bands are going to be those regions where we kind of pointed out earlier that there are slightly different staining patterns that are associated because either there's only thick, only thin, or overlap. So only thin is the I band, And if you look here, the area where there's no overlap just yet, this is called the I band. Right, so we've got two I bands on either side here. Only thin filaments, no overlap. Then along the center where we actually see overlap. So if you look at this region, there's definitely overlap here. This is called the A band. Okay. And now if you look even closer to the center in between the A band, there's an area where there isn't any overlap, right? So in between just the thick filaments, but it's going to be on the periphery of the M line itself. So this is called the H zone. So this is where we only have thick filaments. Okay. I also want to point out, or insert rather, the titin protein. So remember, we got these stretchy proteins that are helping to anchor the thick filaments and provide this elasticity and support as the sarcomere shortens. So this is titin. Okay. And let me also make a note of what these filaments are. So actin is referred to as the thin filament. Myosin we called the thick filament. Okay. And let's also make a note at the bottom here that one myofibril, so as you pointed out at the top here, one myofibril is just a repeating of this exact same thing. So many sarcomeres are just lining up against each other to form a myofibril. Okay, so this is what the sarcomere looks like. And let's talk about this structure. We're going to pick up and speak about this structure much more the next time we meet, but I'm going to highlight it here. This is the region where we have the cross bridge happening. The cross bridge is the interaction between actin and myosin, okay? That is where we have the essential events that allow the entire sarcomere to shorten. So this is called a cross bridge here. Oops. Okay, super important, the cross bridge. And so we'll pick up this and talk about this the next time. But basically we used to have, oh, we've got a couple minutes, I'm sorry. So basically we used to have this understanding, or we used to think, the earlier physiologists used to think that these two proteins were both moving, that they're both sliding across each other. So the thick filament and the thin filament are kind of sliding across each other. This is what allows the sarcomere to shorten. And as we've kind of understood this process a little bit better, and again, I talked about how difficult it is to actually figure out what's going on because of the way that we have to visualize this microscopically. But what we've really come to understand is that both filaments are not actually sliding, but instead, myosin is grabbing onto actin and dragging actin to the center of the Z line. So myosin is really what's doing the work here. Okay, actin is kind of just chilling out, waiting for calcium to come in. Calcium will come in, bind to troponin, remember that complex of 3 proteins that regulates this whole process. When calcium binds to troponin, it's going to drag tropomyosin off of those binding sites, which exposes those binding sites, such that the myosin heads can now reach up and grab onto those actin site, onto those myosin sites on actin. And then what happens is, because the myosin head is what's being energized, that's where the ATP action is, the myosin is what's doing the work. It's going to grab that actin filament and drag it towards the M line. And that's what brings about the shortening of the sarcomere, okay? So we'll talk about the cross-bridge cycle steps, but this is what cross-bridge cycling is. It's the repeated reaching up of myosin. So myosin reaches up, grabs onto actin and drags actin so that the actin filaments get closer to the M line, whereas the thick filaments get closer to the Z line. And so that's what brings about Z lines coming closer and closer and closer together. And that shortens the sarcomere, shortens the myofibril, shortens the muscle cell, shortens the fascicle, shortens the entire muscle. And that's what we get muscle contraction. Okay. Now we also want to think here that when the muscle contracts, it's really just dragging the tendon, right? The tendon is not contracting. The tendon is the attach point. It's going to be attaching from the origin to the bone, from the insertion point to the bone. And so tendons are just moving, whereas the actual dragging or shortening is being done by the muscle body itself.